Staphylococcus aureus is a gram-positive, cockeye-shaped bacterium that is arranged in clusters. Colonization of human skin and anterior nares is becoming increasingly common worldwide, with estimates predicting at least a quarter of the world's population carry this bacteria. Researchers consider the bacterial species as an opportunistic pathogen, causing disease when it gains entry into the body's deeper tissues. S. aureus infections continue to contribute a significant disease burden to societies across the globe, with its potent virulence and ability to develop resistance to treatment posing particular challenges in our battle against the microbe. Staphylococcus aureus must first gain access to host tissue to successfully cause disease. It utilizes a range of virulence factors to first avoid the immune response and invade host cells, then to cause cell damage and proliferate. These include a large capsule to resist phagocytosis and adhesins including fibronectin and collagen binding protein that will allow it to adhere to host cells. S. aureus can avoid opsonization as it contains a protein A on the surface of its cell wall. Protein A binds to the Fc component of IgG antibodies to inhibit phagocytosis, as the Fab component cannot bind. Furthermore, S. aureus produces invasins to assist it in entering the body. Coagulase is used to convert fibrinogen to fibrin. Fibrin forms clots that S. aureus can use to hide from the immune system. The invasin staphylokinase activates plasminogen to form plasmin, which is able to digest the clots whenever it is ready to allow the pathogen to spread in the body. The enzyme hyaluronidase is produced so that hyaluronic acid can be used as a carbon source, and this leads to necrosis in connective tissue, allowing S. aureus to have a greater ease in spreading through the body. It also produces leukocidins that kill leukocytes. There are also a range of exotoxins that are used by S. aureus in causing tissue damage. Exfoliatins separate layers of the epidermis, causing staphylococcal scalded skin syndrome. Staphylococcal enterotoxin B is the common cause of food poisoning and can potentially cause gastroenteritis. SEB and toxic shock syndrome toxin 1 are superantigens that interact with the MHC class II molecules and T cells and cause the immune system to release a large amount of cytokines that lead to inflammation. A common condition associated with Staphylococcus aureus is the potentially life-threatening toxic shock syndrome, and this is the result of the cytokine release. S. aureus colonies can also produce biofilms, and these have been shown to induce hypoxia in host tissue. This helps colonies grow, as it reduces the efficacy of oxygen-dependent immune cells. Biofilms also provide S. aureus with antibiotic resistance, because antibiotics struggle to penetrate the biofilm. It also has the ability to adapt to different environments and tissues, and responds to external stimuli by upregulating gene products that are beneficial to its survival. A variety of diseases can manifest depending on the location of infection. Upon abrasion of the skin, it is able to enter through the epidermis to the dermis, and the infection will cause a pus-filled abscess to form. Bacteremia is the infection of S. aureus in the bloodstream, and provides a means to travel to other organs. If it reaches the heart, it can cause endocarditis, while infection of bones or muscle can lead to the manifestation of osteomyelitis and septic arthritis. Infection of the upper airway will lead to mucosal lining damage and potentially cause pneumonia. Methicillin-resistant Staphylococcus aureus are strains that are resistant to penicillin and similar beta-lactam antibiotics. This type is common in hospitals and has evolved from the usage of antibiotics, which provides selective pressure and has allowed for colonies to grow. This resistance becomes an issue as it is harder to treat with antibiotics that are generally used to target methicillin-susceptible strains.
S. aureus can be found in a number of places, including the soil, water, and air in our external environment. The pathogen can also be found forming commensal colonies of the skin surface of humans and other animals, with the percentage of the global population persistently colonized increasing with time. Live reservoirs or hosts of S. aureus include our pets, livestock, and humans. However, other common reservoirs include households where food and amenities are shared, fitness facilities often rife with sweat and respiratory droplets across shared equipment, farms, public transport, schools, and fomites, all of which facilitate recurrent infections. So, whilst we know that S. aureus appreciates a good human host, where does it colonise? S. aureus has been found to colonise a number of bodily locations, including the epidermis, armpits, groin area, and the nares, which are one of the most common sites of S. aureus colonisation. S. aureus's passport is fully stamped, displaying a worldwide spread, with most countries having an average rate of methicillin-resistant S. aureus of about 25%, accounted for by both hospital-acquired and community-acquired strains. As with all pathogens, understanding its burden of disease is important to allow for adequate resource allocation. When S. aureus initially emerged as a primary pathogen of human disease, it mostly infected the elderly population, specifically those who had recently been admitted and treated in a medical facility, such as a hospital. Over recent years, the emergence of the community-acquired infections, however, has increased, generating concern within the scientific community. High case numbers of S. aureus infections, accompanied by high death rates in some countries, such as the US, continue to be recorded annually despite growing awareness and treatments. Although S. aureus infections can occur in anyone, a higher disease burden is observed in people on either end of the age spectrum, leaving the young and the elderly especially vulnerable to serious infection, whilst males lose out in the battle of the sexes, being found to be at an increased risk in comparison to their female counterparts. So how does S. aureus spread to others? Transmission can occur via points of direct contact, such as ingestion of contaminated food, or more commonly, contact with an infected discharging wound or inhalation of respiratory droplets from an infected individual. Indirect forms of spread include contact with contaminated everyday objects or medical equipment, such as pacemakers, or vector transmission through pets and livestock. Exogenous S. aureus can gain entry via inhalation or direct contact and then subsequent transfer to the mouth, nose, eyes, or food. Endogenous S. aureus, which usually forms a harmless commensal colony on the skin surface, can gain entry via a breach in the physical defences, imbalance of normal microbiota, or where an individual becomes immunocompromised. How do we diagnose, treat, and hopefully prevent S. aureus infections? When stained using Gram stain procedure and viewed under the microscope, S. aureus will appear purple in colour, consistent with their gram-positive nature, and will be of Koch-eye shape. With conventional agarose gel electrophoresis PCR, often too time-consuming for large sample numbers, and a tendency to have a high false negative sample rate, a more rapid and sensitive technique has been developed. TaqMan PCR is a real-time fluorescence PCR assay which detects several enterotoxin-encoding genes, their products, and the presence of MEK-A gene, which facilitates methicillin resistance, and it does this through the use of primers and dye-labeled probes specific for the target sequence. Reverse passive latex agglutination assay is also another commonly used technique to detect S. aureus infection through the use of polyclonal antibodies specific for the different S. aureus enterotoxins. This technique takes between 2 to 3 days to obtain results and is slightly more expensive than TaqMan PCR, at $12 per strain as opposed to $8. Diagnosis of S. aureus infection via isolation and culture methods usually requires a number of different tests. Commonly, isolated cultures are grown on horse blood agar to monitor for evidence of hemolysis. When contamination is likely, matinol salt-containing agar is used, with the S. aureus giving a positive result. And in addition to this, a catalase test is commonly performed, which tests for the presence of the protein catalase, for which S. aureus is also positive. Antibiotics are the most common treatment of S. aureus infection. However, superbug emergence in S. aureus calls for an ongoing need for the development of new antibiotics. Prior to the emergence of methicillin-resistant strains, methicillin, a beta-lactam agent, was one of the most commonly prescribed antibiotics for S. aureus infection, functioning to disrupt cell wall biosynthesis. Since vancomycin was developed only to see strains resistant to this drug emerge in the late 1990s. Moving forward, a handful of alternative drugs have been designed to treat S. aureus, some of which target membrane potential or protein synthesis. Alternative treatments include vaccines, although their development is made challenging by the extensive array of virulence factors possessed by S. aureus, with previous single antigen vaccine trials being unsuccessful. In addition to vaccines, monoclonal antibodies have also been explored for their ability to disrupt quorum sensing in the pathogen. To prevent S. aureus infections and the rise in resistant strains of the microbe, we turn to effective hygiene practices, a measure which has already been taken with the integration of appropriate sterilisation in healthcare facilities to prevent transmission via medical equipment or implanted devices. Another important measure is educating physicians about the importance of antibiotic stewardship, which aims to combat the misuse and overuse of antibiotics. To win the fight against antibiotic-resistant strains, doctors need to ensure they are prescribing the correct drug at the correct dose and that the patient takes the complete course. This video has introduced S. aureus, how its virulence factors contribute to its pathogenicity, the organism's epidemiology, and its diagnosis and treatment.